Welcome to Unit 4, Lecture 1. In this unit, we're actually going to cover two different chapters. So the first chapter is Chapter 4 that basically covers the cell and the types of cells and the way we use cells, and then Chapter 5 talks a little bit more about the functioning of cells. So in this lecture, we're going to start with lipids, and then we're going to talk about microscopes, okay? There are four main classes of macromolecules. We talked about this a little bit when we discussed photosynthesis and respiration. We were talking about carbohydrates, so we did that already. So in this one, we're going to discuss lipids. Okay, so we're here, and we're going to do the other two in a little bit. So lipids are classified as fats, oils, and waxes. So anything you think of as a fat, all right, wax, candle wax, chapstick, okay, those are all lipids. So they have four general purposes. So we've got waterproofing, chapstick, all right, if you think about chapstick. Chapstick creates that barrier between your lips. Lotion can do that as well, and there are fats in lotions. If you've ever washed your hands after you put lotion on, it feels really weird. Cushioning. In between your organs, there is a fat known as visceral fat. And that visceral fat is actually the fat that's in between your organs. And so they cushion each other, because if you don't have any fat in between your organs and they're rubbing up against each other, it's actually going to get really uncomfortable. You can put holes in them. If you think about friction, if you wear out different seams or anything like that. And then a couple of little different things related to cushioning and waterproofing. We get some insulation. So fat helps keep organisms warm. If you think about whale blubber, okay, the whale blubber is to keep them warm. It does also help with buoyancy and stuff like that, but okay. And then last but not least, long-term energy storage. So if you remember, carbohydrates were energy that your cell was using right then and there to make ATP to do stuff. Fats are also used, or fats are used to store that energy long term. So this is where, if you think about all the different stuff they talk about with diets, as far as carbs being bad, fats being bad, this being bad, that being bad, neither one of them are actually bad. But what happens is, if you have too many calories, your body goes, hey, let's hold on to these. And because fat is a molecule that's easier to store, it basically rearranges it, turns it into fat for storage for later use, and then you can, go into your fat stores and so on and so forth. Just like carbohydrates, they are made up of smaller subunits, but they're not really a monomer and a polymer. Like if you thought about the carbohydrates making a really long chain, they don't really do that in the same sense, but they are made up of two pieces here. We get the glycerol piece, which is kind of like the backbone, and then the fatty acids, which you've got, in this case, we've got three. There are three fatty acids, 1, 2, 3, that actually attach to that glycerol backbone, and that's the basis of a triglyceride. So if you ever hear your parents or doctors or anything talking about triglycerides, that's what they actually look like. And if you have too many of them, yeah, that can be a problem. And we're going to talk a little bit about that. There are two main types of fats, two main types of lipids, I should say. Unsaturated, which are usually plant-based. So think olive oil. Okay, olive oil, canola oil, corn oil, those kinds of things. They generally come from plants, and they have double bonds. So you can see down here, there's a double bond between a carbon atom, and there's usually more than one, but there's at least one. And what that means is they are unsaturated with hydrogens. They don't have as many hydrogens as they could have. A lot of these are usually liquid at room temperature, but then they can also be made saturated by what's called hydrogenation. So peanut butter. Peanut is a plant. Peanut butter should theoretically be liquid, but we hydrogenate it to make it into that spreadable solid thing that it becomes. That is really tasty, but that's what happens. And if you ever see natural peanut butter, a lot of times you'll see it separate and there's like a layer of oil, and you've got to mix it up because it's not hydrogenated. Then we have saturated fats, and saturated fats generally come from animals. So you're talking about your bacon grease, all right? What happens when it cools down? It solidifies in the pan. Your butter, okay? All right, they come from animals. There are no double bonds, so it's saturated at room, or it's saturated with hydrogen. So you can see here this saturated fat. This saturated fat has all of those hydrogens. There's no double bond there. And a lot of times these guys are solid at room temperature. Like I said, your bacon grease, your butter, those kinds of things. All right. There are a couple special lipids that we're going to talk a lot more about when we get into chapter 5, but since we're talking about lipids, we'll bring them up now. Phospholipids are a special kind of lipid that are actually used to build our cell membranes, and they generally actually kind of look like this. Okay, that's what a phospholipid looks like. So they have this head and then these two tails, and they only have 2 fatty acids. And then cholesterol is an important lipid that we use. Number one, it helps us build more hormones. It's the it is the backbone of a lot of hormones, but then also it's embedded in our cell membranes. And that helps that cell membrane withstand those temperature changes. And then if we have too much cholesterol, it ends up becoming atherosclerosis, heart disease, can cause heart attacks and strokes and stuff like that. So we're going to talk a little bit more about those, a little bit more this chapter, but then we're going to talk a lot about next chapter when we talk about the membrane specifically. So membranes, we're going to touch on this a little bit because part of we're talking about is the endomembrane system. So membranes are very important, and they're made of these phospholipids, okay, and you can see here, so they've got this hydrophilic head, and you can kind of see down in this picture right here. They've got this hydrophilic head, so that actually sticks out in either direction. And then they've got these hydrophobic tails, so they've got 2 of them, 1, 2, okay, and they kind of look like a pin, a hairpin, a bobby pin, you know, those kinds of things. All right, and then what they do is they kind of create a barrier, and they create various barriers. So there's the big one that basically surrounds the entire cell, and then in eukaryotic cells, which we're going to talk a lot more about. If you don't remember that word, that's okay, but you are filled with eukaryotic cells. Eukaryotic cells have a lot of membranes inside the cell that is really important for making things happen. So we're going to talk a little bit more about that in the context of the organelles this chapter, but then next chapter we're really going to focus on the membranes. Last but not least, life is really small from the standpoint of what actually characterizes or what is actually life. Life is really small. Okay, so we need to use magnification to see them. So we use microscopes. Right. So microscopes have microscopes have 2 characteristics that we talk about. Magnification is actually making something bigger, okay, bigger, and then resolution, which also talks about clearer. Okay, so microscopes need to balance both of these things. We need to make it bigger, but we also need to be able to see it clearly. So microscopes help us magnify things because when we talk about cells, we're talking about microscopic. What does microscopic mean? It means we need a microscope to see them. Okay, and then we use the resolution to be able to see it clearly. Okay, so when we are looking at these cells in this chapter and then thinking about them working and what they're doing in the next chapter, and as we move through a lot of the rest of the year, all right, we're talking about these cells working on an individual basis, okay, but then working on a collective basis to make the organism function. Okay, and so we use the microscopes to see them, and they need to stay small or else they'd have problems. Cell theory, there's 2 pieces to the cell theories. All living things are made of cells, okay, and cells come from other cells. All right. So you didn't just pop out of the midair, okay? You didn't just come out of nowhere. Cells come from cells. I guess I'll draw an arrow. That's the cell theory. All right. And so we need cells. Cells make up everything. We use microscopes to look at cells because most cells are 1 micron to 100 microns, and you can see this size right here. So lipids, which is what we started the lecture out, is right there. Your individual cells are right there. Eggs, right about there. Okay, and remember, eggs are actually single cells. They're the largest, some of the largest single cells on the planet. And then us. Okay, so just keep that in mind. All right, and they need to stay small or else they don't function. I'm going to talk a little bit more about that in class, so I'm not really going to go too heavy into that. But they need to stay small in order to carry out life's functions. Excuse me. Last but not least, types of microscopes. The light microscope is what we're going to use in class. I'm going to choose a different color because that's not class. Okay, we're going to use the light microscope. Electron microscopes, if you continue to study science and you go into college, there's a good possibility that you will use an electron microscope in college. May or may not happen, but they see a lot more detail. So the big thing about light microscopes, they were first used to identify cells in the 1600s. They've gotten much better. They've gotten much more high tech, but they are still kind of the same. They use light and they use lenses. All right, there you go. We're going to use the compound light microscope, so you'll see that. And it is powerful enough to see a lot of organelles. Ours isn't. For the most part, we will probably see the nucleus, cell wall, cell membrane, and maybe some other little things in there. But those are the big 3 things that we'll be looking at. But the nice thing about the light microscope, and even though they're not as powerful as electron microscopes, the reason we still use them is because of this. Okay, specimens can still be alive. We can look at something under the microscope and actually still see it. We can still observe it living. Whereas with electron microscopes, they have to be killed in order to view it. But we've got 2 types of electron microscopes. The transmission, okay, where we can actually get into the internal structure. Okay, we can actually see the inside of something really well. And then scanning, where we can actually see the external really well. So if we wanted to look at the outside of something, we could use a scanning electron microscope. Okay, so these are the couple of microscopes that we use. All right, your wrap up. Okay, give one specific reason why lipids are important to your body and why you still need to eat fats. Okay, you still need to eat them. Okay, and then put in order from smallest to largest: human, carbohydrate, virus, egg, plant cell, atom. Which one of these is smallest, which one of these is largest, and somewhere in between. And then for these 3 things, which microscope, a light microscope, a transmission electron microscope, or scanning electron microscope would you use to view the following? External structure of the chloroplast, internal structure of the mitochondria, or the paramecium doing its thing in water. Okay, I will see you in class. Have a great day.